Okay. So I'm Margarita Ambrosini. I'm a medical oncology resident at last year in Fondazione Istituto Nazionale dei Tumori, and I work in the GI oncology unit. So I have worked for all of this year in the research field of GI cancers, and my project is focused on a phase 2 clinical trial that we have designed at with my research group at Istituto Nazionale dei Tumori, and is a phase 2 trial with an innovative design that is a window of opportunity clinical trial, so focused on patients and enrolls patients with colorectal cancer that are non-metastatic. So normally these patients go to surgery and possibly adjuvant chemotherapy to be cured. And what we do in this trial is to treat these patients according to their...
Sorry.
Yes?
Could you speak aloud, because we can hear you. It's very difficult.
Okay. I try to change the...
Okay. That's fine.
Okay. Okay, so my microphone is okay. Do we have to restart?
No. You can
okay. Okay. So I was saying that my project is focused on this phase 2 clinical trial. It's a window of opportunity design. So we enroll patients with colorectal cancer that are non-metastatic and resectable to be treated with a short course targeted treatment if a molecular specific alteration is found. Indeed, in colorectal cancer today, the targeted treatment have been studied in patients with metastatic disease, and we know that there are some molecular profiles that are exploited as therapeutic targets. So we know that these treatments work in colorectal cancer, and and the aim of this trial is to look at the activity of this treatment when anticipated in the early setting. So for patients that normally will go to surgery and adjuvant chemotherapy, we do 1 month of treatment. So it's a short course treatment in a window of opportunity design that allows us to possibly observe high responses, because we do a molecular pre-screening to the patient that is centralized at Istituto dei Tumori. And if one of these alterations is found, we expect a high response to these treatments, because we know that tumors in the early stage are more homogeneous because they've not undergone to previous treatments, so they've not undergone the selective clonal evolution and pressure from the previous treatments. So we expect a high activity and possibly a benefit for patients, but this gives us the unique opportunity to have biological samples before and after that this specific treatment has been applied. So the project, the specific project with which I'm applying is based on the translational analysis of this trial, and we will collect tumor samples, blood samples, so we will use them for ctDNA analysis and for the analysis of circulating immunity with peripheral blood mononuclear cells, and also stool samples to look for a potential prognostic or predictive value of microbiota. And this is important because we can really observe in vivo the activity of this treatment in the biological samples of patients, and it will be important to compare the aspect and the molecular profiles of these in these samples before and after that this specific treatment is applied.
Okay. That's sufficient.
Okay.
Question. Okay. Just would you tell us if you have already identified a research line, but among the PhD program, the one which lab would you like to spend and to perform your job?
Okay, so I will continue to work in Istituto Nazionale dei Tumori, and for this project we have the pathology department and also the immunology unit and the target unit that will collaborate to the project and for the analysis of this project. And we also have a partnership with the Human Technopole, that is an external lab, and so I will perform the analysis of these.
Who will be the supervisor of this project?
Is Filippo De Bro, that is the director of the oncology unit in Istituto Tumori, and
sorry, could you repeat, Filippo?
Filippo De Bro. is the chief of GI of oncology in Istituto Tumori, and he was our director of the residency program.
Okay, but sorry, but he's in the list of the project of our PhD school.
Maybe not, but do I have to choose one tutor from that from that list? Okay.
Yes, the professor should be one of the one who works in the PhD program. Yeah.
Okay. So I will have to look at the list again, I think, because I'm sorry, I didn't understand to choose one specific from the list.
Yes, you should apply to one specific research line of a PhD program.
Okay.
So, or at least after, if you are admitted, your professor have to speak with one of them. Okay.
And when should I do this choice?
After, if you are admitted, they ask you. Okay.
Okay. Sorry, but I didn't find specific information about this on the call. I'm sorry. Maybe I didn't understand.
Thank you so much.
Thank you.
Giuliano Federica. Okay.
Hello, can you hear me? Yeah. So my name is Federica Giuliano. I'm a medical oncology resident working at European Institute of Oncology. I'm actually in Gustave in Villejuif doing my external period of residency. In Gustave is a major oncological center in Paris. And I'm actually focusing on breast cancer. My main project is focusing on the study of hormone receptor loss in metastatic breast cancer. This means that we have a huge cohort of 100 samples made of 100 patients, in which we are studying how hormone receptor is lost. This means that we have matched samples from the same patients of primary and metastatic tumor. The primary tumor is a luminal tumor. This means that the tumor expresses hormone receptor, so is hormone receptor positive and HER2 negative, while the metastasis is triple negative. This means that the metastasis is hormone receptor negative, progesterone negative, and HER2 negative. These are samples from the same patients. This means that during the metastatic disease, the patient loses the hormone receptor expression, and actually we do not know why. We know that this kind of thing happens in 5% of the cases, less than 5% of the cases, and it can be related to the therapy, because the patients that expresses the hormone receptor are treated with the endocrine therapy. So we may suppose that the endocrine therapy selectively selects in a Darwinian way the cells that do not express the hormone receptor. On the other hand, we do not know if this is driven, the losing the hormone receptors is driven by either a genomic profile that is alterated, or either a transcriptional profile that is alterated, or either to immune related features that may change between the primary and the metastasis. So what we are actually doing, this is a collaboration project between the Gustave Roussy and between the European Institute of Oncology. We are trying to start from the beginning. So we are trying to look out if there are genomic characteristics with whole exome sequencing of FFPE samples to compare the genomic profile in the primary and the genomic profile of the metastasis. This means that we will compare if there are mutations that are different, if there are some alterations that we may find in the primary and not in the metastasis, and vice versa. But actually this is the start of the project, and this is the start of the first year of the PhD. In the second year, we aim to validate our findings in external models, or also to perform model analysis, like, for example, in artificial intelligence.
No, you can go ahead.
Like artificial intelligence analysis on the images and on the actually on the virtual images of the lanes and the slides of the pathological sites of the primary and of the metastasis. This will be the long-term project in terms of planning. But if we are focusing on what I will do in the first year, in the second year, in the third year, in the first year, I will perform the genomic analysis, like the WES, so the DNA extraction and the actual whole exome sequencing. In the second year, I will perform the bioinformatic and biostatistic analysis. We aim to have the primary results. In the third year, we aim to have the definitive results and to have the manuscript finalized in order to make a publication for the end of the third year of PhD.
Okay, that's fine. Thank you so much. Have you identified a research line in the list of a PhD program?
Yes, there is the Professor Nicola Fusco that is the chief of the pathology anatomy in European Institute of Oncology, that is part of the project.
Okay, thank you so much.
Thank you.
Ascenti Veglio?
Sì, buongiorno.
Good morning.
I will introduce myself first. My name is Velio Ascenti. I'm a resident in radiology. I'm a last year resident, and my focus is about interventional radiology, which is a field of radiology in which we do procedures with imaging guidance that can be ultrasound or CT or cone-beam CT. And with these new therapies that we perform, there are a lot of combination with other specialties and a lot of combination with other fields. And for example, for targeted therapy and ablation therapy. And in this regard, I prepared a project which makes possible a combination and studies the immunomodulation in patients that have metastatic unresectable colon liver cancer with unresectable liver metastasis, colon cancer with unresectable liver metastasis that performs a microwave ablation, which is a procedure in which a generator of microwaves is percutaneously inserted into the liver and ablate an area of the liver with the intent of destroying the tumor cells. It has been shown with several case reports in the literature that it is possible to have a regression of distant metastatic site just with the ablation of one zone. This is in general for all kind of ablation techniques, and this is called the abscopal effect. So it means that it has been.
Sorry, can you hear me?
Yes.
Because everything is frozen. We can't see you speaking.
Okay. But can you can hear me, but you can't see me?
Yeah.
Yeah, now it's working.
That's probably is working now. Okay. Try to go on. We will tell you if something is wrong.
Oh yeah. Okay. Yeah. Sorry for that. I was telling you about the abscopal effect. So in in brief, it means that when someone performs a thermal ablation technique, an ablation technique of a metastatic site, it has been shown that other metastases shrink in different location, and with this phenomenon, it has been thought that ablation technique might generate an immunomodulation effect on the immunomodulatory system. So my project is about trying to see if microwave ablation, which is one specific kind of ablation technology, can actively augment the immunomodulation system. And in this, in particular, we wanted to see if the immunomodulation can be done more strongly when the ablation is associated to chemotherapy, because it has also been seen that some chemotherapeutic drugs like doxorubicin can augment the immunomodulation of the modulatory system. In order to do that, I would like to perform a randomized study with two arms. In one arm, only chemotherapeutic agents, and in the other one without liver metastasis, and in the other one patients with liver metastasis.
Okay. Oh, okay, Federico, yeah, that's sufficient. Okay, good.
Quick question. When talking about randomized controlled trials, there are several features that would be nice to summarize. For example, the presence of blinding and primary and secondary outcome, and so on. Could you briefly summarize one of these features for the first trial you are going to do? For example, what's the main outcome variable for the first trial?
Okay, so
of the treatment.
Yeah, of course, the main outcome would be to see if microwave ablation induces immunomodulatory cell death in the colon rectal cancer patients, which I might not have been able to explain to you what ICD, immunomodulatory cell death, is, but it's inside the abscopal effect that I've already told you, and I don't want to be too. I would like to stay concise in order to.
Okay, thank you.
And the other primary endpoints are to see if
okay, that's enough, Avelio. Have you identified, have you decided which line, which research line would you apply for the PhD program?
No, not yet.
No, not yet. Okay. Thank you so much. Islam, I can see you.
Yeah, yes. It's MD Muhammadul Islam.
Okay. Okay, good morning. 5 minutes even for you to introduce yourself and present your project.
Yeah, thank you for giving me this opportunity. I am MD Muhammadul Islam. I'm a PhD candidate for University of Milan. Now I will. So give me time so that I can ready my presentation.
No slide admitted for presentation. You just have to introduce and speak about your project.
Okay, yeah, sure. So my project topic is actually assessment of the role of chaperone-associated autophagy in resistance to treatment in glioblastoma. So I actually choose this topic because I've been really wanted to work with cancer So that's why I choose this topic. And the main objective of my topic would be like analyzing the contribution of chaperone-associated autophagy in conventional like temozolomide treatment resistance in glioblastoma, and also modulation of chaperone-associated autophagy targets and how they impact on different pathway as well as work more efficiently. Nevertheless, the study of post-translational modification on identified motifs and their elimination or creation can act as favor on CMA without showing any side effects. And the total expected outcome would be of my project would be we'll understand the correlation between chaperone-associated autophagy and metabolic conversion in GBM cell, beside how these changes control therapeutic responses. Also, identification of biomarkers related to CMA along with predictive value of these markers that impact early treatment response. Nevertheless, evaluate miRNAs as well as nucleic acid-based treatment, which can regulate CMA activity. Is there enough, or should I say more? I'm really sorry, I couldn't.
No. If it's okay. If you have finished, that's fine. Have you identified the research line in the PhD program that where would you like to perform your project?
Sorry, can you repeat it again?
Yes, of course. Have you identified a research line within the PhD program where would you like to spend perform your project?
In Italy.
Within the research lines of the PhD programs.
Is that mean like you send the Google sheet in?
You can find it in the website, okay? And you should identify supervisor to perform your project. You can do after, don't worry.
Oh, okay. It's all right, yeah.
Just next one, Saki Sahar. Saki Sahar.
Hello, Professor.
Okay, sorry.
Good morning.
Hello, good morning.
Thank you for granting me the opportunity to interview. My name is Sahar Saki. I'm from Iran. I graduate of a master's degree in genetics. Additionally, I have written paper in English on therapeutic approaches to cancer. During my undergraduate and graduate study, I practiced and carried out several research experiences. I first began my research experience in my university and investigating the effects of sex hormones and cytotoxicity colon cancer cells, and my thesis is about effects of diphtheria, herceptin, immunotoxin, and cytotoxicity in breast cancer cells. And my molecular lab experience is about PCR cloning, Western blot cell culture, and another molecular test in a research laboratory. In addition to laboratory research in the field of molecular genetics, I also have experimental research in the field of clinical experience. I work in a hospital laboratory and collaborated with a research team on cancer samples for patients. The topic of my research project is about proteomic and myelomic investigation of senescence-derived exosome to identify circuits modulating cancer chemoresistance and tumor microenvironment. And I choose this topic because we recognize that there are still scientific gaps in this area, including limited information of senescence-derived exosomes and their impact on the tumor microenvironment and the molecular circuits and this project has three objects. First is identify protein and mRNAs in senescence-derived exosomes that may play a role in to chemoresistance. Second is investigate the effects of these exosomes on cancer cells and the tumor. And finally, aim is identify molecular circuit and signaling pathway regulated by senescence-derived exosomes. In this project, in methodology is first of all is a cell culture step. In the cell culture stage, selected cells and cell culture, and after that the medium then centrifuge at low speed to remove cells and large particles, followed by filtration through small pore filters to eliminate remaining cell debris and smaller particle. And in the next stage is we use ultracentrifugation stage to condition medium to centrifuge at very high speed in place the exosome at the bottom of the tube. But how to this particle we know is a real exosome. We use characterized test with NTA technique and electronic microscopy. Nanoparticle tracking analysis or NTA method is used to determine the quality and quantity of exosomes. This technique measures the size and number of exosomes, helping to assess their size, distribution, and quantity in the sample. Additionally, electron microscopy to employ to observe the precise structure and identify exosome in the microscope level. In this method is in examining the morphology and confirming the presence of exosome in the sample. The second part of a research project is about proteomic study. First step is protein extraction. We handle the sample by treating the isolated exosome, and this process including lysis to exosome to release the protein, followed by their purification and concentration. Then we use an enzyme to digest the protein into peptides. Next step we use in mass spectrometry method for proteomic analysis. Protein are first digested into.
Okay, that's enough. Thank you.
Yes.
What about the research line? Have you already identified the supervisor and lab where?
Yes, I interested about this project, and I interested in research microRNAs and predictive biomarker for neurodegenerative disease. It's about Don Carlo Gonocchi Foundation, I think.
Gonocchi.
Yes.
Thank you.
You're welcome.
Okay.
Yes, good morning, ma'am. Okay, my name is Syed Hasnain Raza Shah. I'm from Pakistan. I have done my graduation as Doctor of Pharmacy, a 5-year graduation course from Bahauddin Zakir University, Multan, and later on I have done with my Masters in Biochemistry and Molecular Biology from University of Agriculture, Faisalabad. And after my graduation, I just got a job in a public sector forensic laboratory, which is an ISO 17025 ratio 2017 accredited, as well as it is accredited. It is pre-qualified, WHO pre-qualified laboratory, a state-of-the-art my laboratory of my country. I was searching for my PhD, my doctoral study, in order to for my capacity building in my organization. Also, my organization also support me to improve my education So I got admission at Zhejiang University, Hangzhou. So I got admission there in 2020, and at that time there was pandemic and conditions, right? So I was unable to move there. But soon after, when the pandemic goes on and went on, and the restrictions were waived off, I moved there in 2023, and I worked there for about 1.5 years, and I have and the meantime when from 2020, from the year 2020 to year 2023, I have completed my coursework online, and I completed my coursework. I have a transcript of this. Then just after entering the mainland China, I just attended my lab and started work there. My key work was to synthesize the antibodies against acute lung injury caused by COVID-19 SARS-CoV-2 virus. So I worked there, and we performed research to engineer an antibody against SARS-CoV-2 lung injury. And also I have published a research paper. If you will require, I will show you here. I can share you the screen if you ask me, if you agree with me to share this. In my research work, we started with the immunized and mouse model, and then immunized with an antigenic. We immunize our mice with genetically engineered receptor binding domain of S1 protein of virus, Crohn virus, and then antibodies were produced in the mice, and we detect this by ELISA, and after the third or fourth immunization dose, the titer was the highest of the ELISA, antibody. Then we extract the RNA from the spleen cell of that mice, and then we quantify and qualify the RNA from ELISA techniques and also from some UV techniques. Then we found the cDNA from the extracted RNA, and we also identify the genes from which the antibodies against the immunized receptor binding domain was produced. Then we also segregate the genes, and we found the gene encoding the variable length low variable length chain and variable length chain of heavy chain gene, and both of the genes were linked together with a suitable cysteine and glycine.
You that's enough. Thank you so much. Have you
Sorry, it's remaining to. I have to discuss too much things.
Oh yes, but you have no more time. Everyone has just 5 minutes.
Okay. Yes.
Okay. Okay. Have you already identified the research line? A supervisor?
Yes. I would like to do research in the validation and innovative biobotanic method for the differential diagnosis of chronic pulmonary diseases, as I have experience in lung injury, and my research project could also easily be modified against lung cancer and other lung diseases and infective lung diseases, because we just need to find an antigen against which we can.
That's enough. Okay, thank you so much.
Welcome, madam.
Torelli. Maria Fortunata, she's online? Yeah. Can you hear us?
Can you hear me?
Yes.
Good morning. I'm Maria Fortunata Torelli, and I recently completed my master's degree in medical biotechnologies.
where I focused at University, where I focused my thesis on the investigation, the role of adipose-derived mesenchymal stem cells in supporting the progression of endometrial cancer. This experience enhanced my skills in molecular and cellular biology, as well as different lab techniques. During my Erasmus program, I gained experience in biology and immunology, working with techniques like ELISA, protein detection, isolation, and treatment of bacteriology. So, this international experience enhanced my skills, enhanced my adaptability in a diverse research environment. In addition to my academic background, I gained experience from my fellowship in biomedical research at University. There I was involved in sample collection and processing, data management, and collaborative project focusing on aging and its related diseases. My passion for cancer research started during my bachelor internship. During my bachelor internship, I focused an experimental research aiming at improving pharmacological treatment of elderly patients. And I'm skilled in data analysis tools like GraphPad, Excel, ImageJ, and I started learning R programming for statistical analysis. And I'm enthusiastic about the chance to advance my research in glioblastoma and CMA as reported in my research proposal, titled Studying Treatment Adaptation and Resistance in Glioblastoma through CMA, aim to understand the role of chaperone-mediated autophagy in glioblastoma. The goal is to understand how CMA contribute to metabolic reprogramming and treatment resistance by identifying target and possibly developing a biomarker that can predict treatment responses, possibly contribute to personalized medicine. Chaperone-mediated autophagy is a selective form of autophagy that selectively degrades protein that contain a specific consenting segment that is key PR2-like motif that is recognized by a chaperone HSC17 for lysosome degradation. But it has been reported in literature that CMA contribute to metabolic reprogramming through post-translational modification. So, because this post-translational modification can expose or obscure this motif. If the motif is obscured, the targeted protein is not degraded through CMA, altering protein turnover and cellular processes. So I aim to identify this target by using quantitative proteomics in syngeneic cells by using both sensitive and resistant cells. I aim to culture these cells and induce autophagy through nutrition deprivation and pharmacological treatment. Using techniques like immunoprecipitation to identify by using antibodies that able to against key components of CMA, such as HSC17 that recognize the motive for the degradation. Other techniques will be mass spectrometry, bioinformatic tools for the analysis of the data collected and identification of molecular pathway involved and proteins targeted by CMA, with the aim to have a comprehensive understanding of the role of CMA at molecular level. Finally,
Okay. That's enough. Your research line?
Biochemistry of cancer.
Sorry?
Biochemistry of cancer.
Okay. The supervisor would be?
Luisa Ottobrini. Professor Luisa.
Thank you so much.
Thank you so much.